Let's talk about the procedures that is going to be performed for chemical sequencing method और मैक्सिमम गिलबर्ट मेथड। इस प्रोसीजर को हम दो हिस्सों में तकसीम कर सकते हैं। एक हिस्सा बेस मॉडिफिकेशन का है और दूसरा हिस्सा जो है वो स्ट्रैंड डैमेज का है। जो वेरिएशंस आती हैं वो बेस मॉडिफिकेशन वाले हिस्से में आती हैं, बिकॉज डीएनए के 4 डिफरेंट किस्म के न्यूक्लियोटाइड्स को मॉडिफाई करने के लिए हमें डिफरेंट कंडीशंस, डिफरेंट केमिकल्स की ज़रूरत पेश आती है। जबकि बाद में जब हम डीएनए स्ट्रैंड को प्रोसेस करना चाहते हैं फॉर स्ट्रैंड डैमेज और स्ट्रैंड ब्रेकेज, दैट कैन बी डन इन द सेम वे। वी कैन प्रोसीड फर्दर यूजिंग दी पिपरडीन। बेस मॉडिफिकेशन के लिए 4 डिफरेंट किस्म के रिएक्शन को सेटअप किया जा सकता है। नॉर्मली रिकमेंड किया जाता है कि 4 रिएक्शंस किए जाएं, ए रिएक्शन, ए प्लस जी रिएक्शन, टी रिएक्शन एंड टी प्लस C reaction. Again, sometimes some researchers also establish the 5th one, that is A more than C reaction. So what we actually need to mix up for base modification can be seen in this table. In this table we can see that we have the steps, the components. We need the modifier, base modifier. For guanosine or A and G we need dimethyl sulfate here. Then here we need piperidine for A plus G reaction. For C plus T reaction we need hydrazine, and we proceed further. We have hydrazine for C reaction in the presence of sodium chloride. जैसा कि पहले जिक्र हुआ था, सोडियम क्लोराइड की मौजूदगी में हाइड्रोजीन इज गोइंग टू रिमूव साइटोसिन, is going to modify cytosine with more efficiency. Then we have to follow these steps. Sonicated DNA is going to be added in equal volume. Radiolabeled DNA is going to be added as recommended, 5 microliter, 10 microliter, 10 microliter and so on. Then these all 4 tubes, they are going to be chilled at 4 degrees centigrade in the on the ice, or we can place them at 0 degrees centigrade. And subsequently we have to follow these compositions and the centrifugation series, and then how to handle the supernatant, because some supernatant have to be separated on a waste separate appropriate waste bottle. Some can be discarded in the general way. Once these modifiers are added, then we have to go through a series of steps that involve the centrifugation as well as lyophilization. Once we are through with these steps, that is the addition of the base modifiers and the addition of different reagents as well as incubation or centrifugation steps and removal and disposal of the supernatants after centrifugation, then we have to pass through a series of centrifugation as well as lyophilization stage. This is a list of the steps, like we have to add sodium acetate, ethanol, keep it at minus 70 degrees centigrade, centrifugation, removal of the supernatant, addition of the ethanol, and centrifugation again, removal of supernatant, lyophilization, and by this way we are going to proceed further. The lyophilization is basically a method that is going to remove all the liquid component from the tube, leaving behind the solid component, that is the DNA. By this method we successfully modify the bases. Now the DNA has been modified in 4 different tubes at different nucleotides. The next step would be to proceed further for strand damage, and for that purpose we are going to resuspend the lyophilized DNA sample in the DNA base modified DNA by vertexing in the 1 molar piperidine solution. This piperidine solution is basically going to perform the cleavage at sugar and phosphate level, ultimately removing that particular modified base, and the strand is going to be broken down. For this purpose, once we mix it, piperidine is added into the tubes. We are going to incubate it at 95 degrees centigrade. After vertex mixing, we are going to incubate it at 90 degrees centigrade for 30 minutes. The temperature is high, the incubation period is longer, so there are chances that the lids of these tubes, they can pop up, they can get open. So either place some weight over these lids or use the round bath rack with a screw down pressure plate. That will ensure that the tube lids, they are not going to pop up and the radioactive material is not going to be distributed or disrupted in the surrounding atmosphere. After the incubation period, we are going to make a cooling at room temperature. All the tubes are placed at the room temperature to get cooled down. The lids are opened and they are covered with the parafilm. The needle is used to make holes in the parafilm, and these tubes are subject to rotary vacuum evaporator. The purpose is to remove the last traces of the piperidine, because if we don't remove the piperidine, ultimately it is going to go towards the gel electrophoresis and it is going to interfere with the result of gel electrophoresis. So after the tubes have been through the rotary evaporator, we are going to remove the tubes, discard the parafilm. We are going to add 20 microliter water and we are going to vertex mix the DNA for 30 seconds so that the DNA gets dissolved in that particular volume of the water. अब इस स्टेज के ऊपर अगर हम चाहें, हम इस सारे प्रोसीजर को एक दफ़ा फिर रिपीट कर सकते हैं so that we can get rid of the excess of piperidine or the last traces of the piperidine. In the meantime, we have to ensure using the handheld counter whether there is reduction in the radioactivity or the samples or the walls of the tubes are still heavily giving out the radiations. Once again the process of rotary evaporator is and the lyophilization is repeated, and for that purpose we are going to subject this evaporation step for 15 to 30 minutes, and the step number 6 and 7 is repeated unless we are ensured that the radiation has gone down, which can be determined by the handheld scintillation counter, handheld equipment to determine the radiation. That is Serennikov counting apparatus, handheld, and then we can proceed further to study the DNA samples by sequencing gel electrophoresis. For that purpose, we add sequencing gel loading buffer into each tube. We heat the tubes for 1 minute at 90 degrees centigrade so that the DNA samples can be denatured, and then we analyze the sample using the electrophoresis, polyacrylamide gel electrophoresis.